So let's start with chapter 3. Chapter 3 is cell biology, and quite a bit of chapter 3 is actually a homework assignment that you have on your Connect page. It's also linked through Blackboard. There is no option of doing the chapter 3 homework assignment. It is part of your grade, so you have to do it. Please make sure that you get it done before the first exam because it's a percentage of your exam. The parts of chapter 3 that we're going to cover as a class are actually the ones that are the most relevant to human anatomy and physiology. So, terminology. Terminology is a big part of this class, okay? Terminology is going to help you to kind of learn shortcuts on how to define things. So, the first word here is intracellular. That prefix, intra, means inside. There are certain proteins that are intracellular, meaning that you never find them outside the cell, you only find them inside the cell. When somebody has a heart attack and there's damage to their cardiac muscle tissue, they actually look for really specific intracellular proteins because they know that they shouldn't be in the bloodstream. But when cardiac muscle cells die, all of a sudden they see these levels that shouldn't be there. So that's the prefix intra. Extracellular. Extra means outside. Think of the word exit. So extracellular means it's the things that you find outside of the cell. For us, because our body is made out of organs that are made out of cells plus the packing material that surround them, extracellular material may be things like collagen fibers or elastic fibers. It may even be things like ground substance, the extracellular matrix. So things that aren't normally found inside the cell. Intercellular. So INTER. This stands for between. So again, if you think of the human body as being this whole mass of cells that all have to work together, you do have to have communication between those cells. Your body isn't just one cell working by itself. Everybody is dependent on everybody else, so there have to be ways of intercellular communication. So chemicals that get released by one cell go across the body and interact with another cell. That's intercellular. It's between the cells. Now, on the surface of our cells, we have these things called glycoproteins and glycolipids. Glyco means sugar. Proteins means, of course, proteins. Glyco means sugars. Lipids means lipids. Okay? So, glycoproteins, glycolipids. Why would those be important? Well, on the surface of your cell, you actually have these combinations. And they help to identify the cell. They help to identify it as something belonging in the body or not belonging in the body. As well as that, just the fact that you've got this combination of a sugar and a protein or a sugar and a lipid means that it ends up having a longer shelf life in your body. You may have sugar enzymes that break down sugars, but they see a sugar protein and go, wait a minute. That's not just a sugar. I'm going to leave that alone. You may have a protein enzyme, but when it runs into a glycoprotein, it doesn't just see protein, which means that it'll last longer over the long haul. Now, membrane potential. We talked about that word potential. It could, but it isn't necessarily doing something, right? So, membrane potential happens when inside of your cell, so let's say this is a cell, I have a certain number of ions. Let's say I have 10 positive ions inside my cell, but I have 100 positive ions outside of my cell. Is there a difference between what you see inside versus outside? Obviously, 10 versus 100, that's a big difference. We say that that difference between what is inside versus what is outside is the membrane potential. Now, why do we say it's a potential? because it does have the potential to change. I can actually move some of those ions if the conditions are right. So we say it's a membrane potential. That's the charge on the membrane, but it can potentially change. Now, looking at this picture at the top, this figure at the top, you'll notice that I've got my phospholipid bilayer right here, but there are things sticking up past that phospholipid bilayer. See these things here? They're actually extending higher than that. Those are called the glycocalyx. Glycocalyx is a collection of those glycoproteins and glycolipids that we were talking about, as well as carbohydrates that lies on the outer surface of our cell. It lies well, it's basically outside of the most outer portion of our cell. So let's talk about those membrane lipids. We kind of touched on phospholipids and we kind of touched on cholesterol in the last chapter when we were just in general discussing lipids. But let's talk about that in a bit more detail and how it's relevant to our actual cells in our body. So, phospholipids. These are the lipids that looked like this. They had a head and they had two tails. Okay, and you can actually see them here in this, of course, better drawn picture. Phospholipids have a very important characteristic to them. They are what we call bipolar. Okay, as far as lipids in the membrane go, this is the predominant lipid. This is the one that you are going to see the most often. The head of this is hydrophilic. Philia, that means loving. Hydro means water. So the head of this phospholipid absolutely adores water, loves being around it, doesn't mind it being near it, kisses it when it sees it. It's fantastic. But the other end, notice the word bipolar, right? So those two tail ends that you see here, they're hydrophobic. Phobic means fearing, hydro means water. So the tails don't like water, the heads do. So when it comes down to it, that characteristic actually gives us a bit of an advantage. Because when you look at this cell membrane picture over here on the left, you'll notice that the heads are facing outward. So this is the outside of my cell. And they're facing inward. This is the inside of my cell. And then look where the tails are. All of the tails are huddled in the middle, basically preventing things like water from just willy-nilly going in or out of this membrane. If water tries to bypass it, the tails go, ugh, get away from me, I hate you. And they don't let them go by. So it's kind of a way to make sure that the volume in our cells stays fairly stable. Now, a lot of people have a hard time visualizing what this would look like. I want you to picture a basketball, okay? And let's pretend that if I can cut a basketball in half and it stays its shape, okay? If I touch the outside of the basketball, the surface on the outside of the basketball, I'm touching heads. I'm touching phospholipid heads, just like in the picture up here. If I go along the inner surface of that basketball, okay, I'm still touching heads. Admitted, it's the ones that are down here, but I'm still touching these heads. If, on the other hand, so here's the basketball, you look here, in between these two layers, this would be where those tails would be located. That's why I say they can control what goes into or out of that cell, because literally they're all around it. it's not like they're only in one region. The other major lipid that we have inside of a cell membrane is cholesterol. I know you guys are going to probably think, oh, cholesterol, that's bad. Well, yes and no. Cholesterol can be bad if you have too much of it, but we still need it. So in this case, if you look here, see these little things here? These little yellow things? These are actually cholesterol. Cholesterol, like phospholipids, are also bipolar. They have one part that is water loving and one part that is water hating. So this ring structure up here is actually water hating. It's hydrophobic. This stick coming off of the other end is actually water loving. So it has that same bipolar nature that we see with phospholipids. Now, if you look at the picture up here, you'll notice that those rings are hanging out with the tails of my phospholipids. Remember, the rings are hydrophobic. So it makes sense. They would be with the tails because they don't like water. Meanwhile, this stick portion over here is actually sticking in between the heads. That's why you don't see it sticking up over here. It's actually embedded in the other direction. Now, something that's kind of interesting with phospholipids actually have quite a bit of movement to them. I always think of balloons on a swimming pool. If you hit the water, they jiggle around and move around, right? But you can't have a membrane that is that unstable. So let's say that instead of just free balloons on a swimming pool surface, I take huge pieces of duct tape and I kind of tape islands of balloons together. And that makes them a little bit more stable. Will they still move if I hit the water? Yes, but they move more like a unit than independently. That's what cholesterol does. Cholesterol helps to kind of stabilize that phospholipid bilayer a little bit. Does it prevent it from moving at all? No. But it does kind of make it move more like islands instead of independent units. So there is a phrase that they use to describe a cell membrane. It's called the fluid mosaic model. Fluid because it moves. It is fluid, it is ever changing. Mosaic because you've got different parts and pieces in different places. So, as I said, the lipid bilayer is mobile with things floating around in it. So, let's go back to our swimming pool with our balloons. Everybody knows what the little floaties are, right? You've got the unicorn floaty, you've got the one that goes around your waist with the dragon head, you've got the pool noodles, right? Now, imagine me putting those in between the balloons. Your cell membrane has phospholipids, but in between those phospholipids, you have these proteins that are also kind of floating around. So, what's the consequence of this mosaic and fluid nature of a cell membrane? One, it's important in the molecule distribution on the membrane. If I need something to be on the lateral side of my cell, I can physically move it over there. It would be like somebody wearing the little floaty with the dragon head around their waist and walking through the balloons to get to the edge of the pool. I can move things when I need to. Two, slight damage can be repaired because the phospholipids will actually cover it back up. The nature of the phospholipid with its hydrophobic tails and its hydrophilic heads means that when you disrupt them, the tails want to get away from water as quickly as possible. The easiest way for them to do that is just to reform into the way that it was before. And finally, C, it enables two different membranes to fuse together. Literally, if you've got membranes right next to each other and you want to make one humongo cell, all you have to do is kind of flip those phospholipids out and they take on their internal tails, external heads, structure, and you can fuse those membranes together to make one large membrane.